In the previous video, I have told you about the basic structure and concept of prokaryotic cells and eukaryotic cells. In this video, I will tell you the structure of prokaryotic cells in details. So let's start. So the today's topic is structure of prokaryotic cell. You all know that prokaryotic, the term comes from the pro means primitive and karyon means nucleus in Greek word. So prokaryotes are microscopic unicellular organisms. It is microscopic unicellular organisms of relatively simple construction, characterized by the lack of a definite nucleus and membrane-bound intracellular organelles. The prokaryote cells are most primitive types of cells, which are called primitive cells. Prokaryotic cell, like bacteria, does not possess well-formed nucleus. Cell organelles like mitochondria, ER means endoplasmic reticulum, Golgi bodies are absent there. The cell wall is made up of mucopeptide and peptidoglycan. Only 70 ribosomes are scattered within the cytoplasm. So the definition of prokaryotic cells looks like that the most primitive type of cells which are devoid of a true nucleus, well-defined cytoplasmic organelles like mitochondria, endoplasmic reticulum, Golgi bodies, etc. are known as prokaryotic cells. In the previous video, I have told you a definition of prokaryotic cells, that also right, and this also. So the main topic of our this video is the structure of the prokaryotic cells. Now we will move to this. Structurally, it has three main regions: appendages or appendages. It means in the form of flagella and pili or femri. The second one is cell envelope. That means it consists of a capsule, cell wall, and the third one is plasmamembrane, cytoplasm, containing DNA, ribosomes, and other inclusions. And most of the functions of mitochondria, chloroplasts, and Golgi apparatus are carried out by plasma membrane. The detailed structural organization is as follows. We will discuss now here. So first of all, we will discuss about flagella. So what is flagella? Flagella's filamentous structure may be polar or peritrichous. Its diameter is about 20 nanometer. So it is filamentous. Structure. Its diameter about 20 nanometer. A motor apparatus consisting of several distinct proteins, a system of rings embedded in The cell envelope, the basal body, and hook-like structure near the cell surface rotates the flagellar filament. The rings are divided into two main sets. The innermost rings, located in the plasma membrane, comprise the motor apparatus, while the outermost rings, located in the periplasm and outer membrane, support the rod where it is joined to the hook of the filament on the cell surface. So the flagellar ring are divided into the two main sets. The first one is innermost rings, which is located in the plasma membrane, while the outermost, or the second one, is outermost rings located in the periplasm. Its function is flagella help in locomotion. So the second structure of Pugli cells is the fimbriae or pili. Fimbriae and pili are short, hair-like proteinous structure. It also thread-like or hair-like, present on the cell surface of gram-negative bacteria. So it is a too short, hair-like or thread-like proteinaceous structures on the cell surface. So the pili are less in number and are mainly used in sex reduction and serves as a receptor for certain viruses. So the pili less in number used in sex reduction and serves as a receptor of virus. And the fimbriae are also surface projection its diameter is about 3 to 20 nanometer and length is about 0.5 to 20 micrometer. They are large in number and are used to attach the cell with its. So here the function is looks like act as an organ of adhesion or attachment and may form braids during conjugation. And now the cell envelope. Plasma membrane, cell wall and capsule constitute cell envelope. We are discussing about capsules. Capsule. Most prokaryotes contain a gelatinous layer of polysaccharide or polypeptide layer at the outside of the cell wall called capsule. So the capsule is a gelatinous layer of polysaccharide or polypeptide. Sometimes a thin layer of polysaccharide is observed on the surface of the growing cells called glycocalyx. It is considered as capsule if it is firmly attached to the cell wall or slime layer if loosely attached. Capsule protects the cell from bacteriophages, phagocytes, and lysozymes. It can be a source of nutrients and energy to microbes. It prevents cell from desiccation. So the function is protects the cell from bacteriophages, phagocytes, and lysozymes. Can be a source of nutrients and energy to microbes. Prevents cell from desiccation. So now the next point is cell wall. Cell wall. Beneath this capsule, there is a cell wall that differs in two classes of bacteria. In one class, cell wall shows a thick 15 to 18 nanometer layer of peptidoglycan with teichoic acid. Now the first class. In another class, cell wall contains a thin layer about 10 nanometer of peptidoglycan with teichoic acid. It is followed by periplasmic space outside which there is an outer membrane containing transport protein called podin. Outside the outer membrane, there is a thick layer of lipopolysaccharide. If crystal violet stain is applied over the bacteria and after removal of stain, if the cell appears blue or purple or reddish, then these are designated as gram-positive or negative cells respectively. So, the protoplast of bacterial cell is surrounded by a rigid, thick, elastic cell wall composed of 10 to 20 complex mitogenous material called mycopeptide, lipoprotein, lipopolysaccharide, and phospholipids. So the function gives the shape and integrity of the cell. It protects the cell protoplast from external injury. And now the time is for cell membrane. Cell membrane. The outermost part of the cytoplasm is called cell membrane. This semi-permeable lies just before the cell wall. It is made up of protein, lipid. The cell membrane is semi-permeable outermost part of the cell just before the cell wall. So it shows typical structure as per fluid mosaic model. It is about 5 to 10 nanometer thick and consists of phospholipids 40%, phospholipid 40% and proteins 60%. It lies below the peptidoglycan layer of the cell wall. It performs a variety of functions like osmotic or permeability barriers, transports nutrients and ions, carries out respiratory and photosynthetic roles, synthesizes membrane lipids, murin, secretes extra cytoplasmic proteins, coordinates DNA replication and cell division, performs chemotaxis and sensing functions, regulates many enzymatic activities, helps in signal transduction, helps in cell wall synthesis and septum formation. So it is permeability barriers and transport nutrients and ions. Now the mesosomes. Meso means middle and zoma means body. So the cytoplasmic membrane extends within the cytoplasm of prokaryotes, forming tubular or loop-like vesicular bodies called mesosomes. So it is extends within the cytoplasm, tubular or loop-like structure. This mesosome contains respiratory enzymes. That's why it is often considered as incipient mitochondria. It is also called incipient mitochondria. It is also called peripheral body or chondroid. So the plasma membrane of gram-positive bacteria forms vesicular, lamellar, tubular invaginations into the cytoplasm or form stalks or vesicles attached to the inner membrane surface. These are called mesosomes or peripheral body or chondroid. It contains light harvesting pigments and ATPase. They also help in DNA replication and segregation. Cell wall synthesis. There are two types of mesosomes. One type is septal mesosome. Participate in the formation of cross walls. Formation of cross wall and latellar mesosome. Rich in respiratory enzymes. So the function of mesosomes is look like it takes part in the differential wall formation during cell division. Formed in the replica of DNA, it establishes the connection between the cytoplasmic membrane and nuclear materials. Cellular respiration occurs here. And the vacuoles. A prokaryotic cell may possess one or more vacuole in the cytoplasm. Function is vacuoles help to store the food and gases within the cytoplasm. The ribosomes. The granular appearance of the cytoplasm is due to the presence of free ribosomes. These are of 70s in size. 70s in size, being composed of 30s and 50s subunits. Ribosomes are involved in the process of translation or protein synthesis. And now, nucleoid or NGO. In the center of the bacterial cell, there is a convoluted mass of supercoiled DNA with 10% RNA and proteins, mostly RNA polymerase. Around the whole mass, there is a fine fibrous material but no membrane. It shows a double-stranded DNA. So double-stranded DNA. supercoiled DNA with 10% RNA. And proteins, mostly RNA, polymerase. So it shows double-stranded DNA forming a circular chromosome which is devoid of histone protein. But several basic proteins have also been detected along with the mass of chromosomes. There are several small circular extranuclear bodies containing DNA called plasmids or sex factors or conjugates or extra chromosomal replicants or transfer factors. Plasmids or sex factors or conjugates or extra chromosomal replicants or transfer factors. The DNA of plasmid replicate independently of the nucleoid. They contain about 2% of the total nucleic acid of the cell. Contain 2% of the total nucleic acid. Of the cell. Some plasmids remain closely associated with the nucleoid and are called episomes. Some plasmids remain closely associated with the nucleoid. A plasmid remain closely associated with the nucleoid and are called if we got function here, it is the controlling center of the cell. And now the inclusion bodies. Cytoplasm contains two types of storage granules. Some are enclosed by membrane, includes chlorosomes, contain photosynthetic pigments, carboxysomes involved in the regulation of carbon metabolism, magnetosome contain iron in the form of magnetite, while some are devoid of membranes, includes glycan granules, crystals and paracrystalline granules. So the some inclusion bodies are chlorosome, contain photosynthetic pigments, carboxysomes involved in the regulation of carbon metabolism, magnetosome containing iron in the form of magnetite. And now endospores. These are dormant, tough, highly resistant. These are endospores. These are dormant, tough, highly resistant, thick-walled structure. Thick-walled structure produced by small number of bacteria produced during sporulation. sporulation. They help the bacterium to withstand environmental stress. They occur in soil and water for a long time. It occurs soil and water for a long time. They help the bacterium to withstand environmental stress. They occur in the soil and water for a long time. The position of the endospore may be terminal, subterminal, or central. Lateral endospores are seen occasionally. A thin covering known as the exosporium. A thin covering known as the exosporium overlies the endospore. Endospore usually occurs in Bacillus, Clostridium, etc. And now the cytoskeleton. Traditional eukaryote cytoskeletons have also been found in bacteria and other prokaryotes. FTS, Z, MRE, B, and CRES have been proved to be bacterial counterpart of eukaryotic tubulin, actin filament, and intermediate filaments, respectively. FTSZ assemble to a Z ring at the cell division site. While MREB are involved in maintaining cell shape. Regulating chromosome segregation, where CRES is crucial for cell morphology, maintenance. There are also some prokaryotic unique cytoskeleton components playing crucial roles in cell division, chromosome segregation, and cell morphology. So now the functions. First one is carbohydrate fermentation and absorption. Large intestinal lumen is mostly anaerobic. Here the bacteria turn polysaccharides into short-chain fatty acids by form of fermentation called saccharolytic fermentation, producing acetic acid, propionic acid, and butyric acid, providing the host cell a major source of useful energy as well as helping the body to absorb calcium, magnesium, and iron. Gases and organic acids like lactic acids are also produced by saccharolytic fermentation. Acetic acid is used by muscle. Propionic acid helps the liver produce ATP, and butyric acid provides energy to gut cells and may prevent cancer. Evidence also indicates that bacteria of lipids and facilitate the body to absorb vitamins K. Beneficial flora increase the gut's absorption of water, reduce counts of damaging bacteria, increase growth of human gut cells, and stimulate growth of indigenous bacteria. The second one is trophic effects. The increased growth, proliferation, and differentiation of intestinal epithelial cells. They also alter intestinal growth by changing the expression of sodium plus or glucose transporters. It helps also immunity. The bacteria stimulate the lymphoid tissue associated with the gut mucosa to produce antibodies to pathogens. The immune system recognizes and fights harmful bacteria, leaving the helpful species alone. Gut bacteria express toll-like receptors (TLRs) that help to repair damage due to injury. Toll-like receptors cause parts of the immune system to repair injury caused by radiation. Toll-like receptors or TLRs helps to discriminate between the pathogenic and commensal bacteria. And metabolic functions also occur by prokaryotic cells. The resident gut microflora mediate the synthesis of vitamins like biotin and folate, as well as absorption of magnesium, calcium, and iron. The gut flora play a major role in metabolizing dietary carcinogens. It also helps in allergy prevention. They are also implicated in preventing allergies and overreaction of the immune system to non-harmful antigens. They also stimulate the immune system and train to respond properly to antigens. It preventing inflammatory bowel disease. Gut bacteria also help the system against inflammatory bowel disease. Cancer. Bacteroids and Clostridium are associated with an increase in tumor growth rate, while Lactobacillus and Bifidobacteria are known to prevent tumor formation. Weight loss. It initiates a shift in the gut flora. So bacteroids in the functions of weight loss, the bacteroids increase as weight loss progresses. And next, the qualities. It has been suggested that commensal bacteria are responsible for the development of qualities. So that's it. Here in this video I have made the detailed structure of prokaryotic cells and functions in detail also. I hope this video will help you a lot. In the next video I will try to emphasize on a topic that is a detailed structure of eukaryotic cells. So thank you.